Is the contributions of plant viruses to phytobiome research. And when we put this session together, I think the idea was really to just kind of initiate a conversation about viruses with their contributions in the phytobiome. So the first, without going into too much, taking up too much time, I just want to introduce the first speaker, which is Rodrigo Valverde, and he's going to talk about viruses in nature, persistent plant viruses, and he's from LSU.
Okay, thank you to the organizers for inviting me to give this talk. And I will begin by saying that viruses are everywhere in plants. That's a picture of the Monet Garden. Some of you probably recognize that. I went there a couple of years ago, and many plants are there infected with viruses. Okay, but we are used to, I think this thing is not changing the pointer is working, no? Yeah, okay. Okay, most of all of us are used to see virus symptoms in crops, and that's just an example of some of that. However, when you go to APS meetings, be sure and pay attention, and especially to ornamental plants. It's amazing how many ornamental plants are infected by viruses. This is in Hawaii, by the way. I was going to the beach, and I just saw that beautiful plant there. But if you are Jim Schoelz, and you're walking around campus, you can run into plant infected with viruses as well. Now here's a salvia infected with a virome virus. Traveling to France, here I am I was in Paris there, and I saw this plant infected with a butylene mosaic virus. So, but some plant viruses do not cause symptoms, and an example of that is sweet potato. Viruses also are found in non-cultivated plants, and here's an example of kudzu, an introduced plant infected with tobacco ring spot virus. When I go to national parks, I always pay attention to plants infected with viruses, and here is opuntia infected with the ring spot causing virus. Here's a very nice picture of Diodia. Here is infected with the Diodia vein chlorosis virus, and here is healthy. Very nice interesting picture in the Baton Rouge campus of LSU. However, most non-cultivated plants do not show symptoms. It makes sense, the virus evolved with the plant, and this is a typical example is spring beauty latent virus. This is a bromovirus, which infects this wild plant, spring beauty, and essentially doesn't cause any symptom. Okay, let's divide plant viruses into groups, acute and persistent. Persistent viruses essentially do not cause symptoms, or at least something that we can detect. They do not move from cell to cell. They don't have the movement protein, and therefore they are present in all tissues of the plant, in theory. Okay? They are transmitted only through seed, and more specifically, only through the gametes, only through pollen or the egg cell. Now, this is an illustration of some of the families that contain persistent viruses. Notice that these families, Partitiviridae, Endornaviridae, Amalgaviridae, Chrysoviridae, and Totiviridae, are families that contain mostly viruses that infect fungi. I have a gel there, electrophoresis, that shows, just to show you more or less the size of the genome of these viruses. Persistent viruses infect many food crops, and I want you to pay attention to two of them here, bell pepper and melon. When you eat bell pepper raw or melon, you are consuming relatively large quantities of a persistent virus, dsRNA and RNA, both of them, okay? But they also occur in some major crops such as avocado, common bean, and tomato. They occur also in non-cultivated plants. And recently, we began a survey, and we find that some non-cultivated plants, wild plants, are infected with persistent viruses as well. Now, my hypothesis is that persistent viruses are in a symbiotic relationship with the host. And in many cases, it's a mutualistic interaction, and that gives the plant host an adaptive advantage, maybe possibly tolerance to biotic or abiotic stresses. However, in some cases, this interaction could be parasitic, therefore giving them disadvantage. But in many cases, I think that in many cases, due to modern plant breeding and movement of these viruses in crops, we might be dealing with a commensalistic interaction. Okay, well, I want to emphasize one property of these viruses, because it's very important to understand how difficult it is to work with them, is that they are not transmitted by any conventional methods. So you cannot transmit it, not even by grafting. Only through the gametes, as I mentioned, and overall ranges from 70 to 100% by the XL or the pollen. So in order to work or to conduct comparative studies of these viruses, one will have to select lines of the plant with virus and without virus. So my approach was essentially to look at seed lots and screen many plants and find one that might not have the virus. So that is illustrated here with the endorna viruses of black turtle soup bean. I actually planted many plants up to about 100 of a seed lot of black turtle soup bean. Finally, I found one that was virus-free. I self-it, and that became a line. In the case of bell pepper marengo infected with the endorna virus, this actually was done, something similar, but that was done in the lab of Marilyn Rusink while she was at the Nobel Foundation, and she provided me with these lines. I have two other examples there of other persistent viruses, one partitive virus and amalga virus, in which I follow the same approach. So I want just to show you one slide of the partitive virus, pepper cryptic virus 1, and one of the amalga virus. And just to show you that essentially there was just no difference in the plant phenotype. The fruit phenotype was the same too, and also including the yields. I also inoculated this plant with several plant viruses, these two lines, and there was just no differential effect. Now, in the case of tomato, infected with southern tomato virus, similarly, the plants were very similar. Maybe the one that was negative a little bit slower in growing, but eventually they grew the same. And minor difference in fruit morphology, but that could be due to genetics. There's just no difference between the infected and the healthy. Okay, now let's focus on endorna viruses, which I want to be more in-depth. And these are naked RNA viruses that range between 9.6 kilobases to 17.6, and they infect plants, fungi, and oomycetes. I have here the genome organization of a typical endorna virus. The double-stranded form of these viruses that infect plants contain an interesting feature, a nick near the 5' end. Okay, here is the genome organization of several endorna viruses, including here's one from Alternaria, and the largest one is from Vicia faba. As you notice, they vary tremendously in size. When we do when we construct a phylogenetic tree with these viruses, what we see is something interesting, is that plant endorna viruses do not cluster all together. As you can see, they're mixed with fungal endorna viruses as well. There's a lot of work being done currently to try to reorganize this family. Okay, as I mentioned before, these viruses infect important crops, such as the one that I list here: pepper, common bean, melon, rice, avocado, and barley, and other crops. I have to show a dsRNA gel. Of course, some of you know me that I love to run dsRNA gels. So this is very important because I want to point out something here. Here I have the dsRNA of several endorna viruses and a putative one here, but also showing the DNA of the host. So one thing that is interesting, that most double-stranded ribonucleic acid methods to purify them co-purify DNA of the host, and that's something that you have to have in mind and use DNAs, okay? Okay, I want to focus on our results of work on bell pepper endorna virus and common bean endorna viruses. Common bean, a very important crop, nutritionally and also economically. Many years ago, it was found that common bean was infected by an endonavirus. Even some sequence was deposited in the gene bank. But then later, we found out that by running gels longer, you will see there's two bands. Ryo Okada in Japan, a collaborator, went ahead and cloned and sequenced these two bands, and they ended up being two completely different viruses. About 1 kb difference in size and a minor difference in genome organization. These two endonaviruses infect common bean alone and together, most of the times together. And these include common bean that are cultivated, but also Phytobiome vulgaris that is wild. So let's look at the center of origin of Phytobiome vulgaris, and it's very well established that Phytobiome vulgaris was originated in central Mexico toward the west, and then disseminated north and south. And about 10,000 years ago, common bean was domesticated in two centers of domestication. One is called Mesoamerica, and the other one, the Andean region, independently. Now, we're lucky that we have a germplasm system here in the US that you can access so many genotypes of crops. So we decide to screen some of these materials. When we look at genotypes of Mesoamerican origin, look at the wild Phytobiome vulgaris, we found that about 15% of them were infected with endonaviruses. When we look at land races, about 39%, and when we look at modern cultivars and breeding lines, 93% infected with these endonaviruses. When we go and look at the material from of Andean origin, we notice that you can see there is just we were not able to find plants, wild plants infected with the virus. Land races, we found only 7%, and modern cultivars and breeding lines, only 12% infected. This table summarizes that, and just to emphasize the 93% infection of endonaviruses in Mesoamerica versus the 12% in the genotypes of Andean origin. It's interesting also to notice that mixed infections of the two endonaviruses are essentially more common in Mesoamerica than in the other region. So these two lines of black turtle soup that I mentioned at the beginning that I selected, we began to conduct comparative studies, and essentially, as with other persistent viruses, there was just no difference in the phenotype or the seed morphology. In collaboration with Juan Osorno, a bean breeder in North Dakota State University, he did a cross of two cultivars, Red Hawk, which is endonavirus-free, crossed with Stampede, which contained the two endonaviruses. And then he, of course, being a breeder, they developed plenty of recombinant inbred lines. We look only at 61 of them, and notice that the two viruses are inherited together in 51 of the recombinant inbred lines. Only 6 with one virus, with the virus 2, nothing, and 4 were endonavirus-free. My student, Susara Cancun, we just finished his PhD, did some work on these comparative studies too. And as you can see, there is essentially no effect on grain yield. We did greenhouse and outside experiments on this. We did inoculation of these two lines with different plant viruses, and I'm going to illustrate that only with one of those, some hemosaic virus. Essentially, the symptoms are very similar, maybe a bit more severe on the negative line. However, when we did some qPCR, one thing that you notice is that in mixed infections of the two viruses and some hemosaic virus, there was just sorry about that. There was just not much effect on the gene expression, but look at the levels in a cultivar, Majesty, which infected only with one virus, Phytobiome vulgaris endonavirus 1. Notice the levels of RNA of these viruses, also Closo, a variety infected with one endonavirus only, the levels are high. So we also did RNA-seq on three samples of each line, and you can see here the tremendous variation between the two groups. Now this thing is working, so. However, one thing that we don't like here is that even within individual plants, there is some variation. So, but it's just plenty of differential gene expression between the two lines. In summary, there is no evidence that these two viruses in common bean affect the phenotype or the grain yield. Some hemosaic virus increase the titer of PVE1 and PVE2 in single but not in mixed infections. One thing I want to point out is that earlier domesticators, without knowing of the presence of these viruses in common bean, selected the two viruses for cultivars in Mesoamerica, but not in the Andean region. So it's possible that under some biotic or abiotic stresses, these two viruses could be beneficial to the host, common bean, however, may not be beneficial on the others. They might not be beneficial in the Andean region, but beneficial in Mesoamerica. And finally, the transcriptome analysis show quite a variety of differential expressed genes. Okay, let's switch now to pepper. Peppers, as we know, they are, most of us know capsicum annum, which is a variety of peppers from jalapeno to serrano to bell pepper. However, there are other cultivated bell peppers, and that include bacatum, frutescens, and chanensis, and also pubescens. So we found out that bell pepper and other horticultural types of capsicum annum are infected with an endonavirus, and this is called bell pepper endonavirus. What is interesting is that the virus was found in all commercial cultivars that we have screened so far. So it is universally present in bell pepper. However, the virus is also present in Capsicum bacatum, Capsicum frutescens, and Capsicum chanensis. Okay, the bell pepper endonavirus is a typical endonavirus and also contains, notice the nick in the double-stranded form in the plus strand. Okay, one thing that I did, yes, I did, okay, was develop near isogenic lines, and for that I used the two lines that Marilyn Rusing provided me. What I did is to cross the line that contained the virus with the negative line, and by using the backcross breeding method, I was able to, after many backcrosses, as you can see here, to develop near isogenic lines, which essentially are 99.9% genetically identical, except for the presence of the virus. So I think, in a way, I'm thinking that I transmitted the virus from one plant to another one. So now we can conduct comparative studies, which are more reliable. And as you can see, frustrating is just identical. They're just not different in the phenotype of the plants, healthy and infected. I went ahead and inoculated these two lines with many plant viruses. I illustrate that with two of them here. Tomato spore wilt virus and potato virus Y, and again, the reaction was very similar. They just nothing that is very frustrating again, okay? Finally, I have an isolate from the field I collected a couple of years ago of pepper mild motavirus, and notice that the line that contained the endonavirus react with mild mosaic when this virus is inoculated. However, the line that is endonavirus-free reacted with mosaic and necrosis. That occurred also in the greenhouse. Okay, Cesar Escalante is a new student in my lab, and he is conducting more comparative studies. He began doing many ELISAs. Here is just some of the results. And again, it appears that the presence of the endonavirus suppress pepper mild motavirus, although this is not significant, statistically significant, as you can see there. Yes, he has to do dsRNA, so he has to. So I asked Cesar to do dsRNA extractions, and this is some of the results. Notice that the presence of pepper mild motavirus is right here, this band here, and the endonavirus is on top. So you can see that when alone, pepper mild motavirus without bell pepper endonavirus, the yields tend to be a little bit higher there. But again, nothing significant really. So he began to doing some qPCR too, and we see the same trend. Essentially, it appears that the bell pepper endonavirus tends to inhibit pepper mild motavirus. He's also working on the effect of this virus on yield, and we noted that the line that is infected by the endonavirus tends to yield less. In this case, fruit weight, but also the number of fruits per plant. Now, in collaboration with Noah Sela and Avid Dombrowski in Israel, we have been doing some transcriptomics and analyzing the plants that we inoculated with different plant viruses. But right now, I want you to focus only on these two, and these are the bell pepper endonavirus infected and bell pepper endonavirus free line. Notice, I'm really pleased about seeing this, that this tells me that these are actually near isogenic lines. Look how similar they are. So when we look at the gene expression of this plant infected with the different viruses, we notice quite a bit of differences, and I just want to just give you some numbers here, and you can see that PBY probably the one that had more differentially expressed genes, and these are actually host and virus specific. We're still working on this, so we hope to do more experiment and learn more about what is the effect of these viruses in acute viruses. In summary, as in the case of common bean endonaviruses, there's no evidence that bell pepper endonavirus have an effect in the plant phenotype. Inoculation of these two isogenic lines with pepper viruses did not result in any differential reaction. One thing that we noticed is that bell pepper endonavirus has a negative effect in the titers of pepper mild motavirus. Preliminary experiments regarding the yield, it appears that the virus, the endonavirus, apparently affect the plant regarding the yield, lowering the yields. And yes, and RNA-seq suggests quite a bit of interaction between bell pepper endonavirus and acute viruses. And again, I want to mention this, just like in the case of common bean, bell pepper breeders, without knowing of the presence of this endonavirus for many years, have been selecting only bell pepper cultivars or lines infected with the virus. And I think in my mind there must be a reason for that. Okay, and finally, let me just mention a bit about some work that we just begun on how common these viruses are in non-cultivated plants, in wild plants. And we found out that about 130 plus plant species, we found that about 6% are infected with endonaviruses. And these are just examples of some of them which are infected plants. Here's a table showing that. Rachel Herschlag, a student in my lab too, is continuing this work, and she shows some of this in her poster. Notice that it does not appear to be a trend in whether the plant is introduced or native or families. It just occurs, seems to me, randomly in different plant species. We are characterizing these viruses at the present time. But also other persistent viruses that I mentioned at the beginning, partitivviruses, probably chrysoviruses, amalgaviruses, are present in wild plants, but not at the rate that we see them with endonaviruses. So in conclusion, these viruses are present in non-cultivated plants, and about 6% are infected with endonaviruses, and lower rate, a percentage with other persistent viruses. Okay, the overall conclusions of my presentation is that persistent viruses occur in specific cultivars in some cross species, and they do not appear to affect the phenotype of the plant. In most cases, once a variety or a crop is infected, they are near 100%. So it's very, as you can see at the beginning, it's very difficult to find virus-free seed when you have a seed lot that is infected. Endonaviruses interact with acute plant viruses and possibly with other plant pathogens or herbivores. We have evidence of endonavirus interaction with fungi. which I didn't present that here. In some crops, such as I mentioned before, bell pepper and melon, endonaviruses are universally present. So they just know healthy melon cultivars and they know healthy bell pepper cultivars. The geographical distribution of some endonaviruses in some crops suggests that these viruses might play a role in the host adaptation to the environment. And finally, persistent plant viruses play an active role in the phytobiome, and I have a new name for them, viral endophytes. Okay. Well, I want to thank my collaborators, particularly Seiyo Sabanasovic of Mississippi State University, Rai Okada and Hiro Moriyama from Japan, my students, Susara Cancun, Cesar Escalante, and Rachel Herschlag. Also to Noah Sela and Abid Dombrowski from the Volcanic Center in Israel, other collaborators, Marilyn Rusink, Juan Osorno, Talo Pastor Corrales, and also very important to the USDA National Plant Germplasm System. It's just amazing how easily it is to obtain this material. Some of the work was done with a feasibility grant from US Israel BARD. Thank you.
Okay, we have time for a few questions. Yes, Riza.
Suggestion always is made that somehow these viruses are beneficial to the host. As you showed, there is no difference in yield. Is there any difference in increase in number of the seeds per plant?
Number of seeds? No, we have not. We have no evidence of that. We have not studied that.
And is there any other system that the people have looked at suggesting the number of the seed is increased, so it's beneficial for survival?
Yeah, survival. Yeah, it's a possibility. Yeah.
There is no example of that one.
But no, we have not looked into that.
Thank you.
Interesting talk. Have you tested any of the endono-free, and also endono-virus present forms with abiotic stresses, in addition to the virus stresses?
Yeah. The only experiment that we have done on that is salt tolerance. We did not see any difference. In the case of bean, we used a system to test for tolerance to salt, salinity. It was no difference. Again, frustrating.
So what, just to add to that point,
Yeah.
so in the tomato example you showed in the first presentation,
Yeah.
there was some increase in, looks like establishment in endonovirus pre-plant or one of, it was endonovirus present plant. You didn't follow on that?
No, I'm not. I'm trying to focus on endonovirus. That was an amalgavirus. Yeah, I'm trying to focus on endonovirus.
That seems to be like an endophyte. Okay.
Yeah.
Rodrigo, I have a quick question.
Yeah.
So if you find an endornavirus in your sample, these are also fungal viruses. So how do you know if this endornavirus is infecting a plant-associated fungus or the plant itself?
Yeah, that's one question that we always have when we try to publish something in our papers, is whether the endonavirus is coming from a fungus or from a plant, from the plant. And we have conducted electron microscopy. We never see any evidence of any hyphal or any evidence of a fungus. We have the virus is not transmitted through grafting. You will expect that that will not be the case if it's a fungal virus. The amount of RNA present in the plant are relatively high, higher than infection with regular viruses. That's another thing that, and when an endophyte or a fungus is infecting the plant, the amounts of viral mycovirus are usually lower, way lower. So on top of that, we also, of course, have done PCR using fungal-specific primers, and there is no evidence. So I think there is very strong evidence that they are actually plant viruses and not mycoviruses.
I think that's about it for now. Thank you again, Rodrigo. That was a great talk. Thank you. If you have other questions, you can see Rodrigo at the break. Let's see here. The next speaker is Anna Whitfield from Kansas State University, and she's going to talk about players in the phytobiome, vector-borne viruses, and their interactions with insects hosts. Let's see if I can get this to, there we go.
So I'd like to thank Lucy and Jim for this invitation to speak in this phytobiome and virus session. I think it's quite timely with the launch of this big, exciting initiative led by APS to really look at the entire phytobiome as a way to improve agriculture. And so with this initiative, there's a new journal, and I'm going to be a senior editor with this Phytobiomes journal. And so I want to just encourage all of you to consider publishing in this new journal. And if you have questions about what might fit, because there have been several of those at this meeting, feel free to talk to me or Carolyn Young, or float your idea by us, and we'd be happy to consider it. So it's a really exciting new journal. It's going to be open access. So we're really happy to launch that. And if you don't have a sticker, see me after my talk, and I'll give you one. So everybody's being branded as Phytobiome. So when you think about the phytobiome, I think that a lot of us like to think about what is the phytobiome. And so they've come up with a definition that phytobiomes consist of plants, their environment, and their associated macro and microorganisms. And as Rodrigo pointed out so nicely, viruses are everywhere. So clearly, as you look at the microbe piece here, viruses are a very big part of the phytobiome, as well as the many other interacting factors. And in my talk today, I'm going to be talking about viruses, but also animals, herbivores, insects that transmit viruses. AndLl be touching on some of these other sections here, the omics data, because what we're trying to do is really understand how the virus and the vector interact and then that results in disease in a plant. And then if we can understand how these interactions occur, then hopefully we can disrupt them and prevent virus infection of plants. And that's our goal, to provide a more safe and secure food supply. So that's really the goal of phytobiomes. And so you can see that really it's integration of many different levels of research. And they know that we're at the beginning of this initiative. So a lot of the first steps are just that characterization of the interaction. And like we talked about science to practice, then once you can characterize it, then you can try and disrupt it. So viruses are transmitted by many diverse arthropods, and I just have a few pictures here. Some of my favorites are the mites. Wheat curl mite transmits many viruses to wheat, very important agronomic crop, and thrips vectors. But today I'm going to be talking about plant hoppers and their transmission of maize mosaic virus. So when you think about plant viruses, and we just had that nice presentation on these persistent viruses, and today I'm talking about mainly the acute and economically important plant viruses, and the majority of them are transmitted by arthropod vectors. So when you look at this graph, you can see most of them are transmitted by hemipterans. That would be aphids, plant hoppers, leaf hoppers, whiteflies, psyllids. They're very good at delivering viruses to plant cells, which they need a living plant cell to replicate. And then we also have some beetle transmission, thrips, and then soil-borne viruses that are transmitted by nematodes and fungi and other protozoans. And there are a few of these acute viruses that do not have an identified vector or no biological vector. So plant viruses have diverse transmission strategies, and I'll just review some of the basics of that biological interaction. Basically, we can divide them into two broad categories. The externally born viruses, so those are the ones that are non-persistent or semi-persistent. So they actually attach to structures of the mouth parts, the cuticle, and they just bind there, and then when an insect feeds on a new plant, they are released and transmitted to the plant. So they don't circulate through the insect body. But today I'm going to be talking about the internal viruses. These are the viruses that enter the guts of the insects. Some of them replicate, the propagative ones, and that's what I'm going to be talking about. And then they eventually have to move to the salivary glands of the insect to be transmitted. So these viruses, the propagative viruses, the plant and the insect are hosts for those viruses. So when we think about persistent propagative viruses, they're actually quite different when you look at them from the majority of other plant-infecting viruses. The majority of them are negative strand RNA viruses. So if you look here, these are the the bunyaviruses and rhabdoviruses, then we have the tinui viruses, and then we have a few others. We have the double-stranded RNA viruses, and one anomaly here of a positive strand RNA virus that actually replicates in the arthropod vector. And so one interesting thing about these viruses is that a lot of them actually are in virus families that actually infect animal hosts as well. So these viruses have acquired a protein that enables them to move through plants, so a virus movement protein. And most of the people in this room are well aware of that. So when we look at the propagative virus transmission cycle, we really can start to see how this plays into the phytobiome because it's quite complex. So the first thing that happens is that the insect vectors feed on a virus-infected plant and acquire the virus. And then the virus is able to enter the insect body, it infects the midgut, replicates, travels to the salivary glands where again it replicates and can be transmitted. So, and then the insect, once it has reached the salivary glands, it has to find a susceptible host to transmit it. But when you overlay this with the ecology of the interaction, you can see there's several steps that can be modulated. And these would be influenced by the environment and the interplay between the plant-virus-vector interaction. So one thing that can happen is we know that virus infection can modify plant quality, which impacts vector performance. We also know that visual and olfactory cues from the plant can influence vector attraction. And then we also know that this virus can have direct effects on the vector, and that's what I'm going to talk about today. Is really trying to dissect, like, what is the effect of the virus on the vector. So today I'm going to talk about Peregrinus matus and maize mosaic virus. This is the corn plant hopper. It's an important pest of maize, sorghum, and millet. As you know, those are very important crops for human consumption and animal consumption. And those are the main hosts of this insect, but they can also survive, but maybe not thrive on other grass hosts. So even once a crop is harvested, the insects can move to these wild grasses and persist and then move back into the cropping systems. So they're a really important problem in production. And those people in my lab know that they can really devastate a plant. These insects can kill a corn plant quite quickly. So we have to stay on the people, undergrads in our lab, to keep changing out the plants. So they're really a serious pest. A single female can lay up to 500 eggs. Their development time on maize during the nymphal stages can vary from 14 to 50 days, so environment has a really big impact on the time of development. And then they live for quite a while, so they can live for 30 to 60 days on maize plants. And those development times and lifespans vary depending on what is the plant host. So maize is actually a very good host for this insect. They also have two different forms. They have a winged form, you can see here, and then a non-winged form. And actually the quality of the plant host really dictates which insect form you're going to have. So when the plant becomes less of a good host, the environmental cues trigger the development of the winged forms, which are able to disperse. And then the brachypterous forms of the insect are really more designed for production and fecundity. So they actually have greater longevity and their immatures develop faster. So it's very interesting that they have this dimorphism. And then also we know the environment. When you think about the phytobiome, this performance of the insect and colonization is enhanced on nitrogen-rich plants. So let's talk a little bit about how the virus is transmitted and what the virus is. This is a nuclear rhabdovirus. It replicates in the nucleus of the plant and the insect cells. It's transmitted in a propagative manner. And this virus persists in the vector for its entire lifetime. So this is quite a while that this insect can transmit when you think the adults can live up to 60 days. So that's actually quite a good proportion of the corn growing cycle. So I'm going to talk today about the interactions between the virus and the vector. So here's just a cartoon of a typical rhabdovirus. These viruses have a glycoprotein that projects out of the membrane. And this glycoprotein is involved in attachment and entry into the insect cells. Whether it has a role in the plant life cycle, we don't know, but it is really important in the insect vector because it's the viral attachment and fusion protein. Then inside the virion, you have RNPs, so you have the ribonucleoprotein complexes, and a few copies of the functional polymerase that are important for replication when the virus is delivered into the host cell for these negative strand RNA viruses. And then when some researchers have characterized the route of the virus in the insect vector, because again, the virus is replicating, they've seen two different routes that are possibly used by the virus. So the insect feeds on the infected plant, the virus is acquired, it travels into the alimentary canal, and it actually enters at the anterior region of the midgut, so that would be over here. So when it's first entering the midgut, it is able to enter those cells and start replicating. And we see that for a lot of other persistent propagative viruses. It seems like that anterior region initiating that replication there is very important. So probably some differences in the gut of the insect that are influencing that process. So these viruses replicate in the midgut, and then they actually infect many different cell types, but they've been seen in the hemocytes. Those are the blood cells of the insect that circulate in the hemolymph, so that those are infected by the virus. So that's one possible route of dissemination, but they've also seen it in the nervous tissue and in the brain, and they think that a neurotropic route could be another way the virus gets to the salivary glands. So Higashi and Brisson have done some nice work looking at the ecology of the P. metis MMV interaction, trying to understand how this virus-vector interaction in the plant can coexist and influence each other. And one thing that they found was that more of those winged forms were produced on MMV-infected older leaves. So that could be important. As the quality of the plant is going down, then you have more of those winged forms that can move the virus to a new host for success of the virus. But interestingly, they really don't see an effect of the virus on survival of the insect. And we've done some similar types of experiments and seen similar things where you look here, they have unexposed insects, MMV exposed, and the MMV exposed infected insects. And you really don't see a difference in the lifespan of the insect. But one interesting thing that they did find here was that infected insects reduce the duration of egg production. So we have the same treatments here, but then when you look at the dotted line of MMV infected, you see that egg production actually stops about 5 days earlier than with the exposed and unexposed insects. So there might be some impact of the virus later in the insect life cycle. So we've worked with MMV and P. metis because it's a really great system for exploring virus vector interactions. It's become we've developed some tools that enable us to ask these basic interaction questions that will enable us to then explore the ecology of the interaction as well. So the first off, RNAi worked really well in plant hoppers. And so I just want to show you this picture. This is from a paper in PLOS One, where we actually knocked down this VATPase gene in these insects, and this is the control insect. And so you actually have a phenotype on the wing of the insect. This gene is expressed highly in the wings, and when you knock it down, you see actually altered wing morphology. And when we look at real-time PCR, transcript abundance is significantly reduced. So we can look at gene function in this insect by feeding insects double-stranded RNA or injecting it. It also is quite large, so it's a nice size and longevity for molecular biology and bioassays. And I say that because the other vectors that I work with are thrips, and they're extremely small and not ideal for this type of work. Also done a tiny bit of work with mites, which I know people that work with mites know they're even more difficult than thrips. So there's some very challenging vector systems out there. We also have developed optimized virus infection methods. One barrier that we had when we first started working with the system was a low infection rate. And so when you're trying to look at the direct effect of the virus on the vector, having a low infection rate is a significant barrier. So we've been able to inject purified virus into the insects. And here in this bar graph, you can see we injected with different concentrations of virus, and we looked at virus titer over time. And you can see that that is increasing, and there is a dose response as well. So more virus, you have more replication and higher titer in the insect. So we think that that's also an important factor in whether an insect becomes a vector, is that dose that they're picking up from the virus-infected plant. And that also relates to transmission. So here are 3 different concentrations of virus, and you can see that really with the high concentration of virus, you have the highest rate of transmission. So what we're trying to do is use omics approaches to understand the virus-vector interaction. We want to characterize virus infection using RNA-seq to identify those differentially expressed genes. And then we're also interested in the conserved responses to propagative viruses by comparing to what others have found using similar systems. And then we're interested in direct interactions as well. So we're really interested in the MMV glycoprotein and interaction with insect molecules that could be involved in the transmission process. So when we look at global gene expression in the insect, we developed experiments where we actually expose the insects to the virus. So we gave them a 7-day acquisition access period. So we had 2 treatments, either MMV infected or healthy insects. And actually, after that acquisition access period, we moved them to healthy plants every 3 days to prevent re-inoculation, because we wanted to look at the direct effect of the virus on the insect and not the combination effect of reacquiring virus or feeding on a virus-infected plant. And so for these experiments, we used equal amounts of male and female RNA. We actually selected insects. We measured the virus titer for individual insects and only used those that had a very high titer of the virus. And this was replicated, and then we used standard procedures for sequencing and analysis. So I just want to point out that we actually used the early adult stage. This is a bar graph showing the accumulation of virus in Peregrinus matus over development. So you can see that there's a gradual increase in virus titer over the lifespan of the insect. And so we used the adult stage here, where we had the highest peak titer in the insect vector. So for our RNA-seq analysis, we were able to assemble 68,000 contigs. We had a mean contig length of 1,400 base pairs, and a good number of these raw reads from our library were able to map to the transcriptome. So one thing that we found with our virus infection of the insect vector is actually there were only a few genes, I would say, out of the entire transcriptome that were differentially expressed. And so we were actually quite surprised by this. We thought we'd see more, because if you think about this virus, it's replicating in all these tissues, almost all the tissues except the ovaries of the insect. And we're only seeing a subtle effect on the insect transcriptome. So we found that 61% of the contigs were able to be annotated. Some of the interesting things that we found were that nuclear localized sequences were enriched. And so that's a logical thing to see when you're working with a nuclear rhabdovirus, so that made sense. That was comforting. Some of the other interesting pathways and genes that we saw that were differentially expressed were reverse transcriptase, chromatin remodeling, and RNA processing and transposable elements. So that's an interesting group of proteins to be affected, and so we're really interested in following up on that, because that could be playing into why this virus is not pathogenic to the insect and it's able to persist. And then a significant amount of the proteins really don't have a match, and that's a story that we see over and over again with these insect vectors, is that a lot of the really important genes that appear don't have an annotation. So we need to really work on identifying the role of these proteins in the infection cycle in the insect. So Kate Martin actually has a poster related to looking at the common responses in propagative to propagative viruses in plant hoppers and leaf hoppers. So I encourage you to visit her at her poster 99 later today. But what she's done is she's actually compared what we found to what other groups have found with plant hopper and leaf hopper viruses. So we compared to this work looking at Gramenella nigrifans and maize streak virus, And then also, this is a Fiji virus. So that's another nuclear rhabdovirus, but she also compared it to some RNA-seq work looking at a Fiji virus, a double-stranded RNA virus, in its plant hopper vector. And so when we look at the conserved responses and unique responses, we actually see that only 16% of those 144 differential contigs were shared between the 3 different systems. So there's only really a handful of those sequences that are similar, that are responding. And 56% of those responsive genes were unique to Peregrinus matus. An interesting thing, so when you look at it, we're comparing plant hoppers and then also with a leaf hopper. What we found was that the responses, even though they were to a dsRNA and a nuclear rhabdoviruses, we found more common responses between the plant hoppers, even though they were infected with diverse viruses. And that's probably because they're so much more closely related than the leaf hopper. So we were a little surprised by that, but it also made sense too. So when we look at those, at the identity of the conserved transcriptome responses, we see that protein binding was the enriched category of genes that we saw. And an interesting thing also is that when we look at those 23 transcripts, some of the initial ones we found in our 144 that were interesting to us were also differentially regulated. So again, we have the category of RNA-dependent, reverse transcriptase, and then transposon-related proteins. We're also interested in tight junction. Those are involved in the connections between cells. So could that be playing a role in cell-cell communication in the insect vector? Could be quite analogous to plasmodesmata or something like that we see with the plant infecting viruses. So that's our transcriptome analysis. So we'll be taking that further, looking at function of those genes. So we're using our RNAi technologies combined with our virus injection to look at the functional role of those 23 different proteins. And so hopefully in a year or two, Kate will be able to present on that data. So we have some promising results, but just too preliminary to talk about right now. So we're also interested in interactomics and identifying viral glycoprotein interactions with insect vectors. Our approach with this work is to really focus on the glycoprotein of MMV because it's involved in many steps in the virus infection cycle at the cellular and insect whole body level. And so we're using the split ubiquitin membrane-based yeast-2 hybrid system to identify interactions between MMVG and the Peregrinus matus. And so our bait would be the G protein, and then our prey in these experiments is a cDNA library from all the different stages of Peregrinus matus that we isolated equal amounts of RNA and added them to make the cDNA library. And so this is work by a former postdoc in the lab, Karen Alvear, where she did an intensive screen, and the end result is that we have identified 122 unique proteins that interacted with MMVG. So I think this was a surprising result. This is a really large number of protein interactions to pull out, so you might think that of course, some of these are probably not true interactors. That's the goal, is to sift through and find out what are the true interactors that are binding. But I'll just point out that there are a couple other papers where they've looked at viral attachment proteins and screens against insect vectors, and they're also finding a large number of proteins. So that's actually comforting that other groups are finding large numbers of proteins in these types of assays, because I've seen some yeast-2 hybrid results where you only get a handful of different proteins. Anyway, but what's interesting is that many of these proteins we were able to annotate and find that they do have roles in membrane fusion and trafficking within the cell, endo and exocytic pathways. Those are things that you would think would be really important in entry and exit of a membrane-bound virus. And then we also found that 28% of those proteins were not annotated. So actually a pretty good percentage of those proteins we were able to identify what a possible function would be in the virus life cycle. But an interesting thing, because a lot of people ask this question, is did any of the interacting proteins interact were they also identified in the RNA-seq analysis? And we saw no overlap between those experiments. So definitely is a different class of interactions with what's binding and what's responding in the insect vector. So I just want to talk about a couple of the proteins that we pulled out. Our approach really is to identify possible interactors using yeast-2 hybrid, and then further confirm that the biological relevance of that interaction using co-localization, and we're using insect cells in our experiments, or co-immunoprecipitation, again using insect cells for expression of the proteins. So the two proteins that I'm going to show you today are apolipoprotein. This is a protein that functions in lipid transport. It's also in some systems been found in insect hemolymph, and other systems a role in innate immunity. Another interesting protein we pulled out was cyclophilin A. This is a chaperone involved in cell signaling, but it's also been implicated in virus movement in other arthropod vectors. So with the ludiovirus aphid interaction, Michelle Celia and Stewart Gray have pulled this out of proteomics assays as well. So this may be a conserved type of protein that can be that is interacting in these propagative and persistent viruses as well. So when we express these proteins in insect cells, either with the Peregrinus matus protein being fused to red fluorescent protein and the glycoprotein against GFP, we see that they actually co-localize. They actually have different distributions in the cell. And we've been able to validate some of those interactions also with co-immunoprecipitation. So when we have both proteins expressed, then we can pull them down with the appropriate tag. So we're really excited about these proteins, and we're going to be using RNAi to look at their function as well. So we're really excited because we've identified a suite of interacting and responsive genes in this insect. So we're starting to dissect the interaction between these two organisms. And as we do that, then we can look for ways to disrupt the interaction. So what we found is that there are diverse pathways of importance involved in a nuclear replicating rhabdovirus that were affected, so transmembrane transport, nuclear and membrane components, and then the reverse transcriptase and transposable element-related sequences. And then we found some common responsive transcripts, even some of those in those interesting categories that were shared between various virus vector pathosystems. And so what we're really excited about is that we can now use these proteins as targets for disrupting virus transmission, and that gets to the APS theme for this session, for this meeting, is science to practice. And so now that we're able to really describe this interaction, there are new questions that arise about arthropod-borne plant viruses and how do they fit into the phytobiome. So one question is, what is the extended phenotype of the effect of a propagative virus on its vector? So we've been able to show that there are just a few genes that are differentially expressed in this insect due to virus infection. So how do those few genes affect the insect and the interaction with the virus and then transmission? So that's what we're really interested in, is putting this into the context of the entire phytobiome. And then also important questions for vector biologists are how does the microbiome of the insect modulate the virus vector interaction? So and then if we can identify those microbial components, can we manipulate the phytobiome to target insect vectors to reduce transmission? Some strategies that we could steal from the animal system would be like using Wolbachia to reduce vector competence. So those strategies are actually being used in the field now. So I'd like to thank the members of my lab, particularly Karen Alvear and Kate Martin for conducting these experiments, and the co-director of the lab, Dorit Rotenberg. Then I want to acknowledge our friends that have shared their data with us and enabled us to look for those common pathways. So PEG is here from their 2012 paper, and then the RNA-seq of the other plant hopper. And funding by the very important granting agencies that enabled us to ask these questions. So, and thank you for your attention.
So I think that this talk has generated a lot of questions. We'll take one, just because we're trying to keep on time.
Yes, no, that's good.
So I have, go ahead, Reese.
Is there any data on the level of replication and accumulation of the viruses in the vector? How much duplication is occurring? It has been quantified?
Yes. So we actually have a paper that came out this year about the virus titer over time and accumulation. So we're looking at viral RNA abundance. We're actually not measuring replication per se, but we're actually looking at the titer or the accumulation of the virus. So actually what happens is we see a gradual increase over development, and then it kind of levels off in the adult stage and just persists. So it's not, so it's replicating and just maintaining this infection. It's not getting extremely high in our laboratory conditions, but it's there the entire lifespan of the insect. So it has a great capacity to transmit these viruses if you think they can live for 30 to 60 days and be spreading it in the field.
But
So.
when you compare it with plant, which one is better host?
Oh, which one is a better host? Okay, well, we haven't done a direct comparison between the plant and the insect, but, you know, this virus is pretty bad on the plant, so I think it's very pathogenic on the plant. In some ways, we think of these viruses as a almost mutualist with the insect vector, because a lot of times they make the plant a better host, and we see that with the Thrips hospital-virus system as well. So yeah, it's more of a pathogen of the plant and a mutualist, or just, yeah, with the insect.
Well, I'd like to
Okay.
thank Anna for a great talk. In the interest of staying on time, you have your questions for later. I have a question late, but I'll do it later. And so the next speaker is Sadia Bikal from the University of Illinois. And she's going to talk about characterization of diverse virus populations associated with plant pathogenic nematodes.
Good morning. Thanks, Jim, for inviting me to present today. I'm honored by that. And I'm going to talk today about nematode viruses and some of the viruses that we uncovered through our project over the years. This research was done in Chris Lambert's lab, and I was a member of the team of his lab. As you know, nematodes are a big component of soil and environment. They are very numerous and abundant in all kinds of environment. They inhabit different environments, and they are fascinating in shape and forms. The most important ones, which can account for 50% of the phylum, are marine nematodes. Some of them are animal-associated nematode, which cause diseases. Some are free livings, and you probably heard of Caenorhabditis elegans, which is the genetic model in animals. The plant nematode, which I'm going to talk about today, account only for 10% of the nematode population, nematode phyla. Interestingly, despite the diversity of nematodes in different environments and their abundance, very little is known about the virus associate. So don't take me wrong, I'm not talking about nematode vectoring viruses, but we are looking at more from the perspective of virus infecting nematode and replicating and having an impact on nematode population. In fact, viruses are vectored by nematode, and the first report dates from 1958, and thanks to Les for loaning me the slide from his class. This is a typical viral symptom of the gray fan leaf virus transmitted by Ziphinema index. Some reports have been actually documented sporadically over the half century, but as I said, those are very light reports on nematode and on virus infecting nematode. The first report dates from 1959 when Laurenberg observed a root knot nematode showing debilitating symptoms on the nematode, which seems to be fitting a viral symptom. In 1980s, Pioneer have done a very thorough study on an iridovirus of the insect parasitic nematode Tomamermes gosgrovi. 30 years later, nematode viruses have resurfaced in 2011 by a report of 2 nodaviruses of the C. elegans nematode. And their genomes have been sequenced through that study. Shortly after that, our group have isolated 5 viruses through RNA-seq project on the soybean cyst nematode. So nematode viruses probably have a different ecological niche from insect viruses or plant viruses. It's interesting to be invited today in this session since it's basically it talks about the phytobiome and the contribution of the phytobiome, the viruses on the phytobiome. Clearly, the nematode do have a very intimate relationship with plants, and most of them are underground. They are microscopic. I'm talking about the plant nematode, of course. On the cyst nematode, the life cycle starts with the eggs who are laid on in the ground, in soil, hatching into J2 stages, infecting roots, root tips, and then evolving into adults through 2 molts. Usually females will be attached still to the plants, while males are free and will fertilize the females, and the females then will lay eggs in the ground, and the cycle will start again. So the viruses in this environment are probably going to face several barriers, probably soil barriers, and also we don't know much about other transmission. We barely started studying this environment and finding out about viral transmission and whether whether viruses are transmitted through all life stages and what impact they have in the nematode. So, as I mentioned, the virus discovery has began through a transcriptome analysis of eggs and juveniles of SCN, soybean cyst nematode. The soybean cyst nematode is a very important pest on soybean. It's basically the most damaging pathogen of soybean, and it incurs billions of dollars of damage on soybean. So we got funding to do the transcriptome analysis on the nematode, and it just happened to observe that some of our population have been declining in the greenhouse. So we collected those nematode-sick population, and we extracted RNA and made cDNA, and then sequenced using Illumina sequencing to finally the sequencing resulted into 33,494 contigs assembled, and those contigs were blasted into the gene bank to look at that homology in the gene bank. So as you see below here in the table, oh, I don't know what I did. Sorry. I guess it was him. I'm sorry. I have to. we uncovered diverse numbers of viruses. The first 4 viruses that we found were negative RNA viruses, single-strand RNA viruses, and the homology or the genome was really strong to those viruses. So we were wondering in the beginning whether this is just an error or a fluke or just a contamination on the nematode. Maybe viruses were just surface contaminating the nematode. When we looked at closely, we had clearly had a very good homology of those viruses to different type of viruses. Some of them were insect-related viruses, and others were plant-related viruses. This slide shows the genome organization of 2 of those viruses, the SNV virus, which is a midway virus, closely related to the one of the nematode virus. The RSV from the nematode seems to be closely related to the NCMV virus. And the genome range from 11 kilobase to 13 kilobase. Like 1 year later, we uncovered another virus, RNA virus, positive strand RNA virus, which had the biggest genome, 20 kilobase, and it seemed to have one polyprotein RNA, and we were interested to characterize that one as well. In the beginning, we were very surprised by the discovery, and we went ahead and did some electron microscopy to basically verify that we are finding viral particles on those nematode tissue. Those are sections from electron micrograms from some of the infected females. And as you can see, there is evidence of what you call viral factories, for example, in pollen A, and some buddings of membrane buddings of the viruses. In the lower panels, you can see the viruses are enclosed in some sort of membranes of the cellular membranes of the nematode. This particular virus is the positive RNA virus, which seems to belong to flavivirus. This tree is a taxonomic organization of the nematode viruses. As you can see, the 4 first RNA minus viruses belong to different genuses. One of them belongs to the Rhabdoviridae family, the SCRV nematode. The SCNV is closer to the midway virus, which is a tick virus, basically an insect virus. And we have the SCTV, which is closely related to a tinea virus belonging to the Biniaviridae family. And then and the last one is the SCPV that belongs to Libovirus. So it's interesting to notice that from just this study, we uncovered 4 RNA viruses that belong to different type of genuses and family of the virus world. the nematode virus, that has the positive strand RNA, was very hard to classify. Initially, we thought it was a pestivirus. Then when we looked more at the genome and we analyzed the sequence, it seems to present a methyltransferase enzyme and some subgenomic RNAs, RNA ends, which seem to put it back into the flavivirus genus. So this is a different virus from pestivirus, and we are confident now that it belongs to the flavivirus. One of the interesting study that we want to know is whether those viruses are present in all nematode stages. So we did some RT-qPCR from different juveniles. The table on the top shows some sporadic viruses on juveniles, and then the table below is from male nematode. And those nematode, particularly the male, seem to have higher higher virus load, and that seems to suggest that the viruses may have been accumulating into the adult stage, and mostly the males. That would also suggest that maybe the males play a big factor in viral transmission, and that could be the horizontal transmission is an important factor in nematode-viral interaction. Now, as I mentioned earlier, the nematode has a very interesting relationship with plants. They usually feed in the roots. They develop a feeding cell within a few days after infection. This is actually a root knot nematode, which is a very similar nematode to the soybean cyst nematode. Ways of transmitting viruses may occur during the feeding time when it has been observed that many nematodes may feed from this similar or common feeding cells. On the other hand, we have looked at the plant side, and we screened for many plants, soybeans, to see whether we find any of these viruses in the soybean plants, and so far we haven't been successful, which suggests maybe those viruses do not transmit into the plants or don't cause any impact in plants. So when we started this project, we've been seeing declines in nematode growth in our greenhouses. And the viruses were originated for the transcriptome analysis from greenhouse and lab culture, and we thought maybe this is just a phenomenon where viruses, nematode viruses, happen to be occurring in nematode environment. So we were wondering, and we were interested to look at the field natural population. And we went ahead and sampled many population from soybean cyst nematode population from Iowa and Illinois. And we looked at the prevalence of those viruses, those 5 viruses in those population. One of the region that we had many samples, hundreds of samples, We were collaborating with Greg Tilka from Iowa, and thanks to him for providing the samples. So Iowa was divided into different regions, and we looked at the prevalence of the nematode in those different regions. And as you see from this table, the viruses have different, this is actually percentage viruses determined in 100 samples. So for example, the Nile virus, CNV, seems to be prevalent in 100% of the samples in the northern central region of Iowa. That tells you how important the virus is in that region. While it's totally absent, for example, in the southern east region of Iowa. So that was interesting to observe. Many people started questioning the viral discovery, and we wanted to know whether the viruses had really an impact on the nematode. So we, instead of doing experiment from our greenhouse isolates, we decided to take field population and grow them several generations in the greenhouse and follow up the viral load. And as you see from this table, we determined the viral load using the RT-qPCR through 2 generations of SCN. The numbers you see here are basically differences between the final viral load and the initial viral load. And anywhere where you see an increase or decrease of those viral loads, it meant that initially, for example, an increase of viral load meant that there was no detected viruses in the initial population. Then we had an increase, which is a number that we cannot compare to the initial population. And reciprocally, when you see a decrease of the viral load, it meant that there was a number initially of a viral load that finally disappeared. So throughout this study, we realized that what's happening is the nematode population, when they had an initial viral load that's tolerable, the viruses will grow on the nematode, and later on, when the viral load becomes very heavy, the nematode population will crash and disappear. That's basically very similar to a predator-prey relationship that we see, for example, in these graphs. So the viruses seems to follow, nematode viruses seem to follow this cycle, nematode-prey relationship. So we have seen the nematode impact and the transmission of those viruses throughout the life cycle of the nematode. And we have seen definitely an impact of those viruses on eggs, on nematode eggs, crashing the population in the greenhouses. Those populations are not lab population or greenhouse. They were originated from the field. So one interesting thing we want to look at is nematode mobility and fitness and the impact of the viruses on nematode fitness. So we designed this assay where we made like a scent column just held with the mesh, nematode being able to crawl over the mesh. And basically, essentially, you'll just put the nematode mesh over a cup of water and let the nematode crawl for a few hours. So we designed this experiment, and we were expecting to see some basically increased fitness of healthy nematode versus diseased nematode who had viruses. And so the nematode when inoculated and we put on the top of the column to crawl were disease nematode, and we thought we will uncover mostly healthier nematode. So we measured the viral load at the top and at the bottom of the cap for some of the nematode isolates. And to our surprise, it looks like the nematode, the viral load at the bottom of the cap seemed to have almost 10 times higher viral load than the top nematode. So suggesting that the nematode that were faster to crawl over the column were actually the sick nematode. So this really was very surprising to us, and we were shocked by the result. We redid the experiment several times, and we did this one with the flavivirus nematode. And it seems to be consistent. The same thing happens. So the conclusion on this is probably the virus is giving a selection adaptative, positive selection adaptation to the nematode to reach the plant once it's hatched and at the J2 stage. So that's the only explanation we have so far for this kind of result. So from the biocontrol perspective, we've been thinking, well, since the viruses seem to have impact on nematode, is it possible that those viruses may be used as a biocontrol? Plant parasitic nematode can be controlled with natural pathogen. It has been shown. Bacteria and fungi have been both used as biocontrol agents, but viruses have never been investigated, and we were interested. So throughout our study, it seems to appear that some of those viruses may be used as biocontrol agents. Of course, we're at a very, very initial stage to know, but from our evidence, some of those viruses at least seems to have that potential. So the big question we were asking ourselves is this really specific to the soybean cyst nematode, or are many other viruses out there, and how diverse they are in other nematode other nematode genuses? And one of the similar nematode that actually has a very wide host range, which is a root knot nematode. We decided to do a same analysis, transcriptome analysis on root knot nematode. We took 24 population from root knot nematode. So, and we did similar transcriptome analysis on those nematode, most of them from California. As you see here, this is the life cycle of the root knot nematode. The root knot nematode are very diverse. They have a wide host range, but they have fewer males, and they are mostly parthenogenetic, comparing to the soybean cyst nematode, which is strictly sexual, requires sexual reproduction. So when we did the analysis, which cost us a lot of money, to our surprise, we did not find any viruses on root knot nematode. And when we were really shocked by that. And one question we thought is maybe, although the root knot nematode is very similar to the soybean cyst nematode in its life cycle, the mode of reproduction might be a barrier for the viruses to persist on those kind of nematode. So our goal is to basically stress the nematode, root knot nematode, to stimulate more males. Usually when you stress the root knot nematode, you get more males, and then see if we infect some of the soybean cyst nematode viruses in that way and keep sexual reproduction going, maybe there will be chance for viruses to persist on. But this is all speculative at this point. We just were surprised by the results. In summary, so as you saw, the genomic methods have provided ways to identify nematode viruses. We were surprised that the soybean cyst nematode were basically appear to have a rich number of viruses from diverse groups, while root knot nematode didn't have any. I've seen a poster yesterday where it seems to, though the viruses we discovered have been reported in some of the North Carolina population, and not only on soybean cyst nematode, but also on beet and clover nematodes, soybean cyst nematode. So the ASEAN viruses seems to be diverse and common in lab and field populations, so this is not just a lab phenomenon. Our study indicates that the viruses have been horizontally transmitted by males and vertically transmitted through eggs with the females. The RT-qPCR virus assay shows that the 5 viruses are transmitted independently from generation to generation and seems to have a very negative impact on soybean cyst nematode, which suggests that they are very suppressive to the nematode. We had seen through the eggs and the 2-generation greenhouse assay that there is a correlation of high viral load and ASEAN population crash, suggesting that those viruses may have potential for biocontrol. And not, unfortunately, as I mentioned earlier, not all nematodes have detectable viruses, and maybe at least the one we have looked didn't appear to have any viruses. So that seems to be really interesting. I'd like to acknowledge some of the people who were involved in the project, Costa Balero and his lab, Greg Tilka for providing nematode field samples from Iowa, Chris Lambert for managing the project, and Les Domeyer for helping a lot with, and I definitely used a lot of his viral resources knowledge. Thank you.
Soy, would it, maybe other places they rotate more than just that? Could that be a factor? The amount of crop rotation, could something other than just corn and soy have something to do with it?
Yeah, it's, thanks for the question. It's very hard for me to explain. I mean, crop rotation is definitely practiced in all over the Midwest. Corn, soybean is very common rotation.
I'm starting to see other crops coming in there other than corn and soy, just when we drive around in there. But I'm wondering, if they start diversifying the crops, are you having less of these viruses?
Yeah.
They're suggesting going out in the field and drive around and look, whether that might explain some of this.
Yeah, we don't know so far where the viruses are coming. We definitely know they are underground. That's why the nematode is picking them up. And it's possible with diversifying culture, you have more diversity of you know,
Looking at a very simple thing. Are the areas where you had 90% or 100% just a strict corn soy rotation, and when you get into these other areas where it's very low, are they sticking some other totally different crops in there? I don't know.
yeah. Well, that's an interesting question. I love to know that. That's definitely something to look into.
So we're going to take a break. We're actually going to reconvene back in about 10 minutes at, I believe, 9:45. So if you have more questions, see the speakers during the break or after the session. I want to thank Sadia again for an excellent talk, and we'll be back here in 10 minutes.
question so that it can be heard here and on the live stream. And if you make me nervous, I'll flag you with how many minutes you have left, including your question time. All right.
Well, thank you, Lucy, and also Jim, for organizing this important session. I think today I'm going to talk about a project in the lab, but I want to also start with thanking also my postdoc mentor, Karen Sholtoff. She's not here today. Most of the work I'm going to talk about is a project that we started together when I joined her lab, and then I'm continuing over in my lab right now, and which is basically to understand how viruses basically infect grasses, okay? And Anna mentioned of, how it's important to understand the fundamental interactions that go through, viruses, how they cause disease in plants. And in this case, I'm going to focus on that aspect and how that also, is, important to also translate the findings, is important. The discoveries are important, and then there comes the translation of the findings. So without any ado, so in the audience, we don't need to emphasize on the importance of grasses. They are important because they are sources for our food and also fuel. And so the pathogens that infect grasses, including viruses, are also very significant. For example, wheat streak mosaic virus, in an example, causes severe losses in just wheat alone. That's still an old number. We also heard of some good talks on maize and maize viruses. I was happy to see a good number of talks. And that maize viruses are also important. And so also the viruses of other grasses. However, there is what we think is there is a bottleneck in our understanding of grass-virus interactions at the fundamental level in terms of what are the defense responses, immune signaling pathways that are perturbed by the viruses in grasses when compared to a dicot virus interactions. You can see that, that there is a lot of knowledge in the dicot side, which we could not generalize all the time to a monocot. So our basically work is to essentially bridge this knowledge gap. And in our lab, we are focusing on mainly PMV, which stands for Panicum mosaic virus. It is a positive sense single-stranded RNA virus. It encodes only a few proteins, 6 proteins, and basically it forms a icosahedral virions, and you can see that 30 nanometers in diameter. And one of the interesting features about PMV is it supports the replication of a satellite virus. In this case, it's satellite Panicum mosaic virus, and it encodes only one open reading frame, and that's its coat protein. And it forms about 15 nanometer icosahedral virions. So both of them together infect a wide variety of plant host grasses, and I'm just pointing out a few of them. Some of them are turf grasses, like Saint-Augustine grass, which is very commonly grown in Texas and also Mexico and Oklahoma, I believe. And also it infects field grasses like switchgrass. They're important for putative bioenergy crops. And also it infects proso millets and other millets as well. So millets have been, for us in the lab, the main host plant to study for over almost 15 years during the time when Karen was studying this. They were using this as a host to essentially understand the determinants of the virulence, PMV and its proteins, and SPMV, and how the synergism is working in the plants. So what is still not known is the part of the plant immune system and what is happening to that during these interactions. And unfortunately, the millets and other grasses, the grasses that I just showed you, are very hard to study in terms of the genetics. They are very complex genomes and also larger plants and difficult to grow when compared to a model plant like Benthamiana for dicot viruses. And so we were in search for a sort of, I would say, model plant or grass for studying grass virus interactions. And in about 2010, so I think we came across Brachypodium distichiana. It was, at the time, DOE was putting forward as a model plant or model grass. For those of you who are not familiar with Brachypodium, it's basically a little grass that is in the C3 clade, very close to rice and wheat, but also close to C4 grasses. And so we thought it would be a good one. Important things about Brachypodium is it has a wide range of, community resources that are available, including a sequenced genome. And there were a collection of germplasm, but also a lot of, for functional genomic tools, there is a population of tDNA mutants that we thought would be very useful for our functional analysis. And so we started basically to investigate into whether we can use Brachypodium to begin with for PMV interactions. And I'm going to now walk you through some of the things what we found using Brachypodium as a host for PMV and also some other grass viruses. Okay, so the first thing is we were lucky to see that Brachypodium was a host for PMV, and also, importantly, the synergism that is imparted by the satellite virus. As you see here, basically, you can see PMV causing severe stunting and also chlorosis symptoms. And with the satellite virus, the chlorosis deepens and also even severe stunting symptoms appear. And so this phenotype were almost identical to what we see in field grasses, such as this is an example from an old paper when Dr. Schoelz was characterizing the synergism in pearl millet, and you can see the identical effects of the synergism in field grass and the model grass. So this is very important for us because it kind of gives us confidence in working with the model grass and extrapolate the findings to any other grasses like in field grasses. So the first things we did was, as most of us are already doing, is to look at, we're taking a systems approach to study at the whole genome level what are the gene expression changes that are going through during these interactions. And so for that, we leveraged the resources of Brachypodium. They had a tiling array available early in 2010. We did this analysis and basically looking at global profiling, global expression changes during PMV and SPMV infection. And what was, summarizing the results, some of the things we found, what you are seeing is a overrepresented categories of genes that are induced or repressed during the infection. And there's quite a few gene function categories that are important. And without going into those pie charts, but I can just show some of those here. On your left, you're seeing a transcription factor class called proteins called WRKs. If you are familiar with plant immune responses, WRKs are important transcription factors that mediate transcriptional reprogramming during plant immune signaling. And there are significant induction of these genes during PMV infection in Brachypodium. And also we found almost 25 protein kinases that are misregulated. And many of them are also receptors on plant cell walls and solid cell membranes and could mediate the signaling to the nucleus. And we found some that were downregulated as well, but just to show a few, some of the MAP kinases are also highlighted here that are important for plant immune signaling. And so this gives us some idea of the major gene functional categories that are perturbed during a PMV interaction in Brachypodium. Another way to look at the gene expression profiles is to basically correlate them to plant metabolic pathways. In this case, I'm showing you, we did this analysis using MapMan software, showing you the metabolic pathways of Brachypodium that are perturbed during the interactions. And each of these pathways highlights either a primary metabolism, secondary metabolism, and the little squares in there shows genes that in those specific pathways either induced, identified by red, or green for downregulated. Just to get a quick overview, we did this and we found that several genes in primary metabolism are downregulated, including some lipid genes here, and a lot of photosynthesis and light reaction chains, reaction enzymes are downregulated. Interestingly, a significant number of secondary metabolism genes are induced. So that's one way, another way to look at some of your gene expression data. But what we're interested in is mostly defense signaling pathways, and so we focused our analysis on plant hormones and their signaling and the genes that are involved in plant defense hormone signaling. You may be familiar with salicylic acid and jasmonic acid and ethylene are the major plant defense hormones in mediating the interactions. And what we found was salicylic acid signaling pathway was strikingly upregulated, and with a huge number of genes, orthologous genes that mostly are pulled out from orthologous of Arabidopsis, or even the nearest ones we studied were in rice. And basically, most of the salicylic acid signaling pathway seems to be upregulated. And as this is a commonly known antagonism between salicylic acid and jasmonic acid. We saw quite a few of jasmonic acid and ethylene signaling genes downregulated. Just keep a note on the FAD7 in JA signaling. It's going to come back again. It is downregulated in our transcriptome data. And so to validate some of the gene expression data, we also did some metabolite profiling. This is in collaboration with Eric Schoelz group when he was still at USDA-ARS, and also Sean Christensen. Their group basically quantified the phytohormones and also diverse other metabolites that I'm going to show you. Well, the metabolite profiles correlate with the gene expression changes. As I'm showing you here, salicylic acid is more abundant in plants, and so is also a precursor of salicylic acid, called cinnamic acid, is also present in higher levels. And we found downregulation or lower accumulation of jasmonic acid, and also its precursor form OPDA. Now, their group also, we quantified some other hormones. In this case, I'm showing you abscisic acid. It seems to be a little bit induced. This is an important hormone for plant abiotic stress and such as drought. What we think is appears to be a crosstalk in this case. Induction of abscisic acid indicates that there is a crosstalk in abiotic stress signaling pathways with the biotic stress signaling pathways. We do have some preliminary data, which I don't have, I'm not going through now, but we found some effects of PMV also on drought tolerance, but that's for another day. Continuing on, we also quantified some fatty acids. Fatty acids are important signaling molecules in plant defense, especially insect interactions. And also they feed into the biosynthesis pathways of oxylipins, including OPDA and also jasmonic acid. What we saw was in a PMV infection of Brachypodium, we significant induction in 16-0 palmitic acid and also 18-2 carbon, linoleic acid, but there was a significant decrease in 18-3 linolenic acid. Now, remember I mentioned FAD7 as an enzyme that we saw was downregulated in our transcriptome data. That actually is an enzyme that mediates the desaturation of linoleic acid to linolenic acid. And so it makes sense to us that that was downregulated, so the substrate was accumulating, and then the product was low abundant in plants. So in addition to looking at basically phytohormones and also fatty acids, we also looked at some secondary messengers of signaling. In this case, I'm going to point out to one observation we made was in levels of hydrogen peroxide. In a typical interactions, I want to emphasize one point that this is a susceptible interaction. Okay, so usually during a susceptible or even virulent interactions, hydrogen peroxide is important for hypersensitive response or even causing necrosis. What we saw was something peculiar in PMV. We saw hydrogen peroxide levels appear to be downregulated. This is an assay we do by staining the leaves of infected or healthy plants with a DAB chemical. That brown precipitate indicates higher amounts of hydrogen peroxide. Basically, even if you zoom into these little the Brachypodium leaves at the cellular level, we saw that most of the hydrogen peroxide in a healthy plant was at the cell wall or cell membrane, and which is what we saw significantly downregulated. Now, what is the significance of a downregulation of hydrogen peroxide in PMV infection is a question we are still addressing. But what we have some ideas that perhaps because it is so important for hypersensitive response and necrosis phenotypes, by downregulating, PMV has an edge in the plant to be able to move forward before the plant kicks on its hypersensitive response or necrosis responses. So that's a little bit about some metabolic profiling work we also did. So in addition to looking at global changes in gene expression and metabolite profiles, we also started looking at really where are each of these PMV and SPMV proteins going in the plant cells, and also what are they interacting with. I don't have time, so I'm going to just skim this slide. We did localization of all the proteins, but I'm going to show you two proteins, which are the capsid proteins. And both of the capsid proteins of the PMV and SPMV appears to be localized in the nucleus. And thanks to Michael Goodin's vectors, we used his vectors to do this analysis. Basically, this is an RNA virus. Its life cycle is in the cytosol, but to us, seeing the proteins going into the nucleus indicates that there is a function of perhaps an important function that these virus proteins are perturbing in the nucleus. And we don't know what it is, but it is a peculiar result that we saw. And so based on some literature of other proteins that go through, other RNA virus proteins that also go through nucleus, such as a GRV open reading frame 3, which Taliensis group has shown that it goes to the nucleus and interacts with the fibrillary protein, and that interaction is important for long distance movement of GRV. We think that similar things could be going on. The PMV proteins could be interacting with some of the nuclear and nucleolar proteins too. We're still working on these two hybrids right now on that. Another graduate student in Karen's lab is working on them right now. But we have also been interested in what other things can go on in the nucleus. And as you have already seen that there is a huge changes in transcriptome. But also nucleus is a site for post-transcriptional gene regulation like splicing and alternative splicing. And we had some preliminary data from our microarrays that we saw some splicing factors to be misregulated during a PMV infection. And so we reasoned that perhaps there is more than those, those two splicing factors could be mediating, huge changes in the alternative splicing. And in addition to that, alternative splicing is important for virus interactions. There's not that much known, but the classic N gene that you probably already are familiar with is also alternatively spliced. It's not much known beyond that there is two forms of N gene. So we set out to study alternative splicing changes that are occurring in plants during virus interactions. In fact, so just to give you an overview, there is different types of splicing that can occur in plants. Some of them are listed here, the major types. Exon skippings are the least predominant in plants. This is data compared to mostly dicots and also some monocots as well. And intron retentions are the most predominant alternative splicing events when compared to humans. Now, each of these different splicing types can result in changes in open reading frames encoding different proteins from the same transcript, and that can have an effect on the function of the protein itself. And so we were interested in understanding these changes during a plant microbe interaction, and so we did basically take advantage of some of the latest tools that are there. RNA-seq is useful for doing such analysis, so we went ahead and did that. And cutting down all the long story, 2 years of work and the workflow through the transcriptome and the alternative splicing analysis, what we found was essentially that there is, here I'm showing you a pie chart of the different splicing types that are misspliced during PMV and SPMV infection. So on the top here is the healthy plants. As expected, there were the majority of splicing types that are present in Brachypodium were intron retentions, and the least abundant were the exon skipping type of events. That fits sort of the patterns that we saw in dicots and also other monocots. And during an infection, we do not see a big change in overall ratios of the different types. For example, intron retentions are still the most abundant, and also exon skippings are the least abundant with both PMV and SPMV infections. So we think this is a qualitative effect. So there is not big changes in qualitative effects. But what is different is in each of these splicing types, there is higher number of splicing events. So that's a quantitative effect of viruses, on splicing. If you look at what are those different splicing events that are misspliced in PMV and SPMV when compared to healthy plants, and then annotate those genes based on putative functions, we see quite a few important genes in plant defenses that are misspliced. I'm showing you 3 classes. NBLRR proteins are the classic N gene is NBLRR protein. And most of those R genes seems to be, some of them are misspliced during infection. And also transcription factors and also splicing factors themselves sometimes get alternatively spliced. So we found a significant number of splicing factors to be misspliced. So there's a lot of data in this one slide. Each of these genes could have an impact on susceptibility of Brachypodium to PMV. How is a question that is the hardest part to answer. But we started looking at 1 or 2 genes. I'm going to show you the first gene we started looking at. That's the missplicing of a splicing regulator. Now, the reason we picked the splicing factor is because splicing factors, and when they get alternatively spliced, they influence the global splicing changes. So it's like maintaining the splicing homeostasis. So basically, what is going on in this case is that this splicing factor called SCL33, which has 2 annotated forms transcripts in the Brachypodium reference genome, a primary transcript and an alternative transcript. What we found in our RNA-seq data was a significantly more number of alternatively spliced forms, especially these intron-retained forms right here. And many of these alternatively spliced variants of this factor are also misexpressed during PMV and SPMV infection. They have a dynamic expression or abundance patterns, that's more appropriate. So to corroborate some of this RNA-seq data, we also did some RT-PCRs really to look at the bands. RNA-seq is all numbers, but here we're looking at all the different isoforms essentially through RT-PCR, and we did identify very more than obviously this is our primary isoform right there, and then there is significantly more than I think you can see at least 4 in a healthy plant, alternatively spliced forms of SCL33. And some of those are developmentally regulated. As you see, as the plant ages in a healthy plant itself, there is some abundance of these higher isoforms, longer isoforms. But note what is happening in a PMV and SPMV infection. Many of those longer isoforms, which are, I guess, developmentally regulated, are prematurely appearing. So there is a perturbation in even the developmental regulation of these transcripts in a virus infection. And we also noticed that new ones appearing in infection, like this one right here. This is absent in healthy plants and only present in PMV and SPMV infection. So we also went ahead and cloned up each one of these and then sequenced them by Sanger sequencing and looked at their essentially protein coding capacities. And we noticed that each one of these longer isoforms, because they were having some portions of introns, they end up having premature stop codons and ends up with a putatively truncated proteins. And the full-length protein of this SCL33 encodes these key domains, RRM at the N-terminus and then the SR domain. And in these truncated proteins, they pretty much lose either the full SR domain or portions of RRM domains. So what is the significance of all of this? Well, each one of these little truncated proteins could be acting as a dominant negative manner, interacting with perhaps even the targets of SCL33 or even the wild-type protein. And that can impact the function of SCL33. We don't know beyond that what are the targets of SCL33, but that's the next question we would like to answer. But just to summarize, so far what we learned with PMV and Brachypodium as a sort of model for our grass virus interactions, we were able to put together some of the global changes that are going on in terms of plant defenses. As the virus, PMV in this case, enters the cell, it's recognized by plant signaling machinery through some of the, we call it virus-associated molecular patterns. And plant triggers defense signaling pathways such as SA. And as a consequence of the antagonism, we think there is a suppression of JA and hydrogen peroxide as well. And these responses are mediated to the nucleus and translates into the transcriptional changes in the defense hormone signaling genes. Now, despite this is a susceptible interaction. So we see all these different signaling pathways triggered. They are induced, and still PMV is able to successfully cause disease. So we think there is something about susceptible interactions. They are one step ahead of plant defense pathways. Either it's a premature timing of these defenses, or even somehow they are actively suppressing some of these defenses. Now, we all know about viruses and their proteins. They can do a lot of things in perturbing host cell physiology. And we think some of these viral proteins are also, in this case, PMV proteins, are trafficking to the nucleus and splicing some of these genes that could be involved in plant defense pathways. There is obviously many questions also to study further, and some of them we are interested in is looking at the splicing stories of each one of these targets that we identified, and what is their role in PMV causing PMV virulence. And also we're interested in hydrogen peroxide. Why is it? How is it suppressed, and how does that also translate into virulence of PMV? But in addition to answering those questions, we were also interested in looking at now what's going back to our, all the different grass viruses and also grasses that are important. What is going on in the other systems? And whether what we learned so far in PMV and Brachypodium can we extrapolate to some of the other grass virus interactions. So we basically took a comparative approach here. And we established with 6 more different grass viruses, grass-infecting viruses, barley stripe mosaic virus, brown mosaic, maize mild mottle, sorghum yellow banding, wheat streak, and foxtail. We established basically Brachypodium as well as another grass in a C4 clade, Setaria, where it is as both hosts for all these virus combinations. So the goal was to essentially compare some of the responses we saw in Brachypodium and PMV, and compare in these different viruses and the 2 different grass types. Now the reason we picked C4 grass was, there's not much known about how C4 specialization in plant photosynthesis and anatomies, influence the different signaling pathways. So that's one reason why we chose C4. And also the agronomic grasses that the viruses infect are C4. Anyway, so the systems are already there. If anybody is interested to study Brachypodium or Setaria and these virus combinations, please feel, we could talk, and we could already use. This work is published, so they're easy to infect. And we haven't tested any insect viruses though. That's another next thing. Perhaps anybody wants to study Brachypodium and Setaria are good also models for insect viruses. Now, so basically the summary is that we started looking at one by one the different things that we saw that were changing in Brachypodium. And the first thing we looked at was SA, JA, and ET responses. We took a qRT-PCR approach, looking at marker genes in the different signaling pathways and quantifying their expression. And I want to note that there is some conserved responses. You can see that the jasmonic salicylic acid is generally upregulated in Brachypodium, and also just JA is downregulated in different grass virus interactions. That response appears to be conserved in Brachy and Setaria as well. Now, that's just the global view, but if you look at carefully, there is some distinct responses, like the ERF1 transcription factor right here is induced in Brachypodium, but it's suppressed in it's repressed in here, and then it's induced here. There is a host-dependent responses as changes as well. In addition, we've noted that foxtail and wheat streak, for some reason, they clustered uniquely in their responses. So there is also virus-specific effects in the different responses we saw. So again, there is things we could tease out and study further. So we also started looking at the CL33 regulation or missplicing during the different grass virus combinations. We saw very similar results. This is a qRT-PCR, again showing the different isoforms. Many of them are prematurely accumulating those isoforms, the many viruses, just like PMV did. right here. And, but some are unique only to, while PMV, for example, is able to misplace that isoform that is not present in any of those. So what are each viruses doing to, how they are misplacing uniquely is, again, another question. But, the more questions you answer, the more questions we get. So that's where we are right now to answer also some of these other questions. So wrap it up, and I'll leave. I don't have a conclusion slide, but I want to leave with this slide of an integrated plant immune model that we think viruses are important players in plant microbe interactions. Often when reading reviews of plant immune responses, in all due respect to the authors, viruses are excluded from the summaries, and so we sort of put together based on the existing knowledge at the time, we put together a review of plant immune responses and how viruses cause disease. There's still a lot more to update on this model, but I think in the part of the phytobiome now, being the hot topic, viruses are an important player in this microbe interactions. So with that, sorry for rushing all the slides. I was excited to talk about all the work we did. I can take any questions if there is time. Oh. Totally apologize. I lost my acknowledgement slide there, but this is the most important one. Acknowledge my postdoc advisor and current collaborator, Karen Scholtoff, and also the lab, and a lot of folks who provided us with plant and virus materials and our funding agencies. Thank you.
Unfortunately, we don't have any time for questions, so we'll move along.
Talk later.
And so you can find Kranti. Thank you. And I'd like to introduce our next speaker, who is Michael Gooden, who will be talking about his work on localization and the interactome.
Thank you much. I want to start by thanking Lucy and Jim and the organizers for the opportunity to talk. It's really fantastic to be included in such a wonderful group of speakers, and thank you for your attention today. So this is a new time, new dynamic time in plant pathology, and try to understand how viruses and phytobiomes are going to interact, so to speak. And so I want to lay a few foundational concepts. Let me show you one example, and I didn't bring my slingshot. I'd dearly like to take out that light that's over there. Hopefully you can see the micrographs. But I want to give you, this is one example of a virus assembly site, this green circle here, and it's a virus-infected nucleus. And I want to show you another virus assembly site, and it's this one over here, and it's a kilometer-wide pivot-irrigated crop circle. Yeah? And they differ only in about 9 orders of magnitude, but they are both virus assembly sites. And a comprehensive understanding of virus phytobiome data and concepts is going to have to integrate information from both areas. Yeah? And so I want to address sort of these 2 points in a sort of general integration of how that may play out. That previous talk was absolutely beautiful for setting this up. If we take this picture and enlarge it with this beautiful microscope here, and just look at a single cell, the interactome in a virus-free cell is going to be very different from the interactome in a virus-infected cell, given the fact that viruses dedicate themselves to reprogramming themselves to basically making virus factories. And we see various virus factories here. We've just had a beautiful presentation on the differential splicing, which is going to affect interacting domains. And so protein interactions over here, lipid interactions are going to be very different from over here. So this has to be integrated into the phytobiome concept interaction. Okay, so I want to touch on 4 points. I've sort of channeled the previous speakers putting this talk together, thinking about what they might talk about. And so I want to sort of integrate what I think they might have said, and they said it beautifully, thank goodness. And I want to touch on 4 points. So how different are interactomes between even genetically related viruses? and I'll show you a comparison of 2 strains of potato yellow dwarf virus. And the idea being that if they're different, then viruses themselves provide the probes to dissect cellular functions. So these so-called VAMPs that were just introduced in the previous talk. So that's very important. Then I'll multiply that by those 9 orders of magnitude and talk a little bit about what can viruses inform us about things like climate change, because it's very important to phytobiome research. So I want to talk about the curious case of coffee ring spot virus in Brazil. Obviously, this is an initiative by APS, so what's the relevance of this research to APS? And importantly, the land-grant missions of the universities where we conduct our research. And a few things to touch on there. The question being, must phytobiome research be internationally collaborative? Of course, and I just touch on some dual degree programs and light programs to promote these type of activities. And importantly, in plant virology, we've got to make plant virology exciting to the Pokemon Go generation. And I have some ideas on how we can do that so they don't do it while they're driving. Yeah. So, and then finally, this is driven, obviously, and we've seen in the previous talks, by tools that allow the research to progress. And my lab's made a lot of tools, and what are the new tools that we need? And I feel it's really all about location these days. So with humble respect to my colleagues, I could tell this story with a number of different type of viruses, but I'm going to use rhabdoviruses to tell the story today. So these are negative sense single-stranded RNA genomes, which are encapsulated over their entire length by the nucleocapsid protein. Being negative sense viruses, they encapsulate a RNA-dependent RNA polymerase, an accessory protein. And this nucleocapsid is condensed by the matrix protein. This complex buds through a membrane to acquire a lipid envelope and a glycoprotein. As the name suggests, many of these viruses are rhabdoid in shapes, but interestingly, most of the plant-adapted species are actually bacilliform, so denoted by this dashed line here. So the first talk talked about viruses in nature, and likewise, rhabdoviruses are everywhere. Sort of, if you're more complex than yeast, you probably will encounter a rhabdovirus somewhere in your phylogenetic lineage. I just want to really focus on 2 groups, and these are the plant-adapted species. And I say plant-adapted because in all cases where it's been examined, these viruses replicate in their arthropod vector. So they're much an animal virus as they're a plant, but they replicate in both. We've also started out, I won't show today, but this clade up here is very interesting. So these are rhabdoviruses in phytophagous insects that have not yet acquired a movement protein. So maybe they're just waiting to get that movement protein, and they'll be the next viruses to emerge. So virus evolution, tick-tock, tick-tock, right? Not to confuse you with this slide, don't try to memorize it all. What's important is the beautiful and extreme conservation in the genomes of these viruses. So the nucleocapsid, phosphoprotein, matrix protein, glycoprotein, and polymerase are followed quite strictly in the different genera of these viruses, including these newer emerging viruses that we're starting to describe that are actually bipartite rhabdoviruses. So with rhabdoviruses with a split genome. And that's an interesting evolutionary discussion to be had. You know, why would you split your genome? And we can think about that. Okay, so to get to this idea of viruses coming into a cell and converting that cell to a factory, I'll give you a cartoon, and then I'll show you what that actually looks like. So either via the arthropod or transmission from an adjacent cell, these nucleocapsids have to get into a cell. In the case of nuclear rhabdoviruses, they establish replication complexes in the nucleus, and budded virions get into the perinuclear space and accumulate there. And the cytorhabdoviruses do likewise in the cytoplasm, but nuclear functions, and we just seen in the previous talk, even if you're in the cytoplasm, you affect nuclear functions. Okay, so that's very important. All right, so to show you what it actually looks like by electron microscopy, nucleus in a virus-free Nicotiana benthamiana, and in the case of a nucleus infected with Sancus yellonet virus. And there's this beautiful ring structure, this virus accumulation over here. So that ring, if you express the phosphoprotein as a red fluorescent protein, you see this beautiful ring structure. Likewise in the EM, and in collaboration, in part with Lucy, we've done electron microscopy, immuno-EM to show that these rings are highly enriched, so that light microscopy is not an artifact. We actually do have P-enriched rings. So what's going on at that ring? If we look at the edge of the ring, well, before we get to the edge, that ring is not around nuclei. It's a transgenic plant expressing fibrinogen 1, a nucleolar marker, and the rings do not colocalize. But if we look at the edge of the ring by EM, we see that that's where nucleocapsid condensation is taking place. And then those nucleocapsids will move to the perinuclear space. This is outer nuclear membrane, inner nuclear membrane. And then virus accumulation in the perinuclear space. Okay. So this series of pictures really belies all the other things that are going on. The virus has to compete for translational apparatus. The virus has to shut down defense mechanisms. There's all these physiological processes that are going on in the ability to establish these virus factories. Okay? And so with respect to just virology itself, and then relating that back to the phytobiome, what are these subtle differences in how viruses establish these factories, and what are the implications? So let's look at 2 strains of potato yellow dwarf, and what's very important is to realize that even the structural proteins of viruses, we just saw a case, the capsid protein of PMV goes to the nucleus. So even structural proteins of viruses have functions over and above their structural requirements, right? They do other things. So they're either VAMPs or effectors in some way. Okay. So potato yellow dwarf virus there are 2 strains differentiated primarily by their vectors. One I'll call CYDV, and the other I'll call SYDV, and they're antigenetically can be differentiated. We sequenced the SYDV strain some years ago, and just recently did the CYDV strain. So what's important to know in a pairwise comparison is that these proteins are mostly about 70% identical, and if you allow for similarity, then the similarity gets close to 90%, 80 to 90%. So they're very similar. And for these studies, they are essentially 2 of the most closely related rhabdoviruses that we have to work with. Right? Okay, so let's look at these subtle differences between them. If you do a binary protein interaction and localization map for both viruses, what you see, they're very different, even though the proteins are, in some cases, almost 90% similar. Some of the interactions are conserved, nucleocapsid matrix, NP interactions, self-interactions of the N protein. But for CYDV, many more interactions are present, despite the fact that these proteins are so closely related. And that is also borne out in the localization. So I'll just show you a couple examples. If you do localization of the putative movement protein, the Y protein of CYDV, it clearly targets the nuclear envelope, here the nucleus in red. So it clearly targets the nuclear envelope where the SYDVY protein does not. Another example is the nucleocapsid protein. As a GFP fusion, it's distributed evenly throughout the nucleoplasm in the case of SYDV. In the case of CYDVN, you get a punctate localization and a deformation of the nucleus itself, despite the fact that they're about 80% similar. And so the important concept is then that very few changes are required to influence things like vector specificity, antigenicity, symptom severity, and so forth. And so phytobiome research is really going to have to take into effect virus population structure, variation in viral strains, and so on and so forth. So that's a critical component of studying these interactions at the level of phytobiomes. So what's really interesting, well, at least for me, are what are the functional domains that mediate the differences between these two strains? And I just want to give you one example to try to get you excited about things like that. So, and that will come from the matrix protein of SYDV. So the structural role of this protein is to condense the nucleocapsid during virion formation. But like I said, viruses, viral proteins often have other functions. So let's start very simply. Take the matrix protein, express it as a GFP fusion, and it's targeted to the nucleus in transgenic plants that express histone fused to RFP. So that's simple enough. But if you then target GFP to different membranes in the nucleus, so in the case of tagging the outer nuclear membrane and you express the matrix protein as an RP fusion, there's no change. Well, that's not very exciting. There's no change. But if you target the inner nuclear membrane, now matrix protein goes into the nucleus and so follows the inner nuclear membrane. And if you target GFP to the lumen of the ER, which fills the nuclear envelope, then you fill the nuclear contents with GFP. And this is the real slide. This is not photoshopped in any way, shape, or form. The cells were actually, we've never found an unhappy face. So I don't know if virus plants interactions, they're not necessarily always happy, but in this case they are. And so we were interested in the targeting signal for both nuclear localization and membrane relocalization. So in the wild type case, a GFP fusion target to a plant with RFP targets of the ER fills the entire contents of the nucleus with RFP, right? So the nuclear localization signal is primarily controlled by this KR. You can knock that out, and there's no nuclear localization and no change in membrane relocalization. So interestingly, it's this dileucine. If you mutate that, you have a protein that retains ability to nuclear localize, but does not affect the membranes. And so what's important is that we can uncouple nuclear localization from membrane relocalization. So we're trying to get as, why does this happen? Obviously, the virus needs lipid because it's an enveloped virus. But what is the mechanism and how does it actually get those membranes into the nucleus? So viruses are masters for hijacking cellular functions. And so we asked the question, does this viral targeting signal, is it present in host proteins? And a very exciting find, and we're trying to follow it up, is that the viral targeting signal is present in the highly conserved RAB18 protein. So RAB18 is involved in localizing lipid droplets to the ER. So it's involved in ER maintenance. This is a rhabdovirus. It needs lipids, but it needs lipids in the nucleus. And is the matrix protein involved in relocalizing Rab18 to the nucleus? So that's something we're pursuing. But what's exciting is Rab18 is involved in the assembly of two other enveloped viruses, hepatitis C and dengue virus. So this is quite exciting and something we're pursuing at the moment. Okay, so a very important question for comparing viruses that have similar morphologies and related genetics. Are these functional domains conserved across viral species? And are they conserved in the same proteins? And it's sort of a yes and no answer. So the functions may be conserved, and unfortunately it's just chopped off. But if you could read it, it would tell you that the phosphoprotein of coffee ring spot virus has the same ability to relocalize membranes as the matrix protein of potato yellow dwarf virus. So this is ER targeted to the nuclear envelope, and if you co-express coffee ring spot virus P protein, those membranes relocalize to the nucleus. So coffee ring spot is this bipartite rhabdovirus. It's a different signal, but the functional effect is the same as the matrix protein. But that gets me to coffee ring spot virus, which I'll now amplify my talk by 9 orders of magnitude, and start talking about virus phytobiome interactions at a level of population. So we're continuing the cell biology of coffee ring spot virus, which is basically a bipartite rhabdovirus. So the polymerase is found on RNA2, and the nucleocapsid through glycoproteins on RNA1. And for the coffee drinkers in this room, we can discuss the importance of this virus, but it should be clearly evident by this picture here. These are very interesting mite-transmitted viruses. So we did a survey a few years ago. We were first to sequence this and do the protein localization for this virus. It's a very understudied virus. It flies under the radar of coffee rust, which is the primary concern if you're growing coffee. But we did a survey and we found it on 100% of farms, representing about 75% of the Brazilian coffee growing regions. And what was really interesting in this study, and I don't have time for all the details, is that there was very strong distance relationship. So the distance between collection sites was had the biggest impact on genetic relatedness. So the further away you go, you could walk through this tree. These are the most furthest away collection sites. So that was interesting. But coffee ring spot virus has this really interesting property. And it's not the only virus to show this, but I think it has quite some implications for phytobiome research at the level of ecology and population. And the phenomenon is that, in this case, Chenopodium quinoa is a local lesion host at 24 degrees. And if you incubate these plants at 2 to 4 degrees higher, so 26 to 28 degrees, they become then systemically infected. So a very small increase, 2 to 4 degrees, will cause that plant to be much more susceptible to virus infection. And that just raises all kinds of question for what will climate change do to virus host range expansion or contraction, depending on the virus. And by basically warming plants up by a couple degrees, you can greatly expand the host range of coffee ring spot virus. So it might be a very excellent model sort of climate change related studies. So we're following that up. But there's an even grander scale. So what will climate change mean in terms of virus ecology and virus emergence, particularly in areas where agricultural biodiversity of one meets high biodiversity areas? And the first talk today was absolutely wonderful. You just go out in nature and just start harvesting viruses. And so this is a really exciting time in plant virology. And this is really the domain of metagenomics. And a wonderful review article recently in Phytopath sort of makes the case for this. And put down your Pokemon Go's and just go out into the wild and find these viruses. This is the best time in plant virology since invention of sucrose gradient and infectious clones. This is a fantastic time to be a plant virologist, so go catch them all. All right, so in order to do this research, it has implications. This is not just virus discovery for the sake of virus discovery. This has implications for agriculture. And I'll just use the Brazil case study, but this obviously applies to many countries. So there are 9 crops that represent 90% of the cropland in U.S. and Brazil. These are the 2 world's largest agricultural exporters. So virus emergence in one country has concerns for the other, and vice versa. And we're doing a number of partnerships with universities, and I'd love to talk to people about what type of international collaborations they're establishing, but dual degree programs, sandwich programs, doing workshops in different countries. This is also a very exciting mechanism to engage students in plant pathology and plant virology in particular. Okay? Eduardo's here, glad to see you, he's right here. So dual degree programs, research collaborations. Standing on top a coffee harvester is way more fun than playing Pokemon Go, I'll tell you that right now. But of course, all of this is driven by tools that allow research to progress. And our labs spend a lot of time building tools, marker lines, and vectors, and we've impacted hundreds of papers by now, so that makes me feel very good. But we need more. And boy, Anna just gave me like this Christmas present during her talk, and I want to take you there. Others, of course, are building tools for protein interaction and looking at expression. And the whole point being to get a more and more refined and exquisite definition of all the protein interactions in a cell. So shown here, part of the Arabidopsis interactome. And the question really being, those VAMs, those viral-associated molecular patterns, where do they impact this interactome, and where do they change it? And we've done some of that. All these protein interactions, okay, on a whole proteome or just a one virus case, they all will have to be translated into protein localization. You're going to have to get back into the cell and see what's going on there, right? Regardless of the system. A critical thing to keep in mind is that protein localization in the absence of a virus infection is very different from protein localization in the presence of a virus infection. So look at this phosphoprotein of SYDV. In the absence of a virus infection, it's in the nucleus in these little speckles. In a virus-infected nucleus, it's in these sort of much more punctate, larger areas, and that's the same. That's just one virus. Here's a very, just to kind of really drive that point home. This SC4 interactor 21 interacts with the SYNV movement protein. And shown in red, its localization on microtubules. Okay? What's very interesting is that this microtubule localized protein, when it interacts with the movement protein, actually drives to plasmidesmata. Okay? So it turns out that this interactor is a non-cell autonomous protein, and so the virus may be hijacking it for movement. And we actually did show its interaction with PDLP1, plasmidesmata localized protein. So interaction with viral proteins shifts host proteins. Okay, so the interactome and the localizome in the context of virus is very different than in its absence. And boy, Anna just set this up beautifully in her talk because she's got all these nuclear proteins that are being influenced by virus infection. And how do you know that a protein is in the nucleus? we're still very much dependent on predictive localization, which is terrible. Yeah? So shown here in red are nuclei, and I just walk you through a couple examples. Fibrinogen 1 localizes to the nucleolus, but if you ask the predictive programs, it'll tell you it's in the peroxisome. WIP1 is an outer nuclear membrane. It's predicted to be in the nucleus, but you can't get subcellular localization, right? So yeah, it got it to the nucleus, but it can't tell you that it's in the outer nuclear membrane. Potato yellow dwarf virus, N protein, predicted to be in the cytoplasm. HRP1 goes to nuclear speckles, predicted to be in the cytoplasm. An extreme case, this is a nuclear protein, that SC4 interactor 21 that I just showed you, okay? Not yet. So SC421 tests positive in a nuclear import assay. It's a member of the VOS1 superfamily of cytoplasm-tethered transcription factors. So what we need, this is the next tool that we absolutely need on a whole cell level, but I'm just going to pick the nucleus to start because Anna needs it, okay? And me too. But the plant nucleome, the full complement of proteins that are trafficked to and into and at times out of the nucleus, in addition to those proteins that mediate nucleocytoplasmic transport per se. In the previous talk, we just saw that the cytoplasmically replicating virus sends its capsid protein to the nucleus to do stuff. So it doesn't matter where your site of replication is, cytoplasm or nucleus, those viruses affect the plant nucleome. And I just showed you by those localization, we have terrible definition of the proteins in this organelle. So what we need is something that has been done with human proteins, a protein atlas, okay, for plant and virus interactions. We need to know where the plant proteins are in the nucleus, in the physiologically normal state, and we need to know where they are in the context of virus infection, because their localizations are going to change in many cases, and we have examples of that. So there may be some proposals out there that address this issue, so if you're interested in this, this will drive all kinds of excellent collaboration. So let's talk about that, and let's get these resources. So coming back to my starting slide, yeah, I'm done. So we have our two assembly sites. It's an exciting time. Plant virus metagenomics, plant virus ecology, a huge part of the future of plant virology. And on the cell biology side, we really need these comprehensive cell biology resources to be integrated into the Phytobiome story. So thank you for your attention, and thanks to the folks in the lab. There's at least 6 countries represented in these 5 people. So this is really worldwide collaborative research. So thanks, present and past members of the lab. Thank you.
Unfortunately, we don't have time for any questions, but thank you very much for a beautiful talk. And we're just going to have Jim Schoelz give us a little wrap-up of the session.
First of all, I'd like to thank all of the speakers. I think the talks were really good today, and I really enjoyed them. I just wanted to, when we were putting together this session, we really, as I saw it, I saw really kind of 3 themes developing in this section. First is viruses are ubiquitous in nature. Second is that viruses induce wide-ranging effects on their hosts. And you can see this in a number of ways. So you can see viruses modulate defenses against other pathogens. They modulate the JA pathways, the SA pathways, and that was kind of emphasized by Kranti. They also, though, will modulate the physiology of the host in response to change how vectors will perceive that host. And so that was something that Anna talked about as well. I mean, you could go on and on about all of the changes in terms of how viruses will modulate their hosts. And then lastly, we're just starting to characterize, I think just starting to characterize, the interactome between viruses and their hosts. Now, when I put together this talk, this slide, I actually kind of pigeonholed talks in a specific category, but I'd like to emphasize that really there was a lot of overlap today between categories. I think virtually all of the speakers were talking about interactomes in some way or another. Now, when I was thinking about this over the past year, I was thinking about, I came across this quote, which I thought was really pretty kind of summarized how I think about this today. And the quote, it actually concerns tobacco mosaic virus, but it really applies to all viruses. And the quote is, "It is clear that the genetic information responsible for the phenomenon we think of as tobacco mosaic virus not only consists of the genes carried in the viral genome, but that numerous plant genes are equally important in viral gene functions. These gene products not only allow the virus to replicate, but may affect functions of evolution that determine what the virus is. Even the processes of pathogenesis and resistance appear to involve similarly precise plant interactions. The challenge of the future is to identify the plant genes involved in these precise interactions and understand both components of genetic information that comprise plant viruses." And this was Bill Dawson in Tobacco Plant Interactions, Virology, 1992. So a couple of points that I want to make about this. First of all, if I have ever thought that I had an original thought in my head, well, probably someone has already thought of it. And the other thing is that I think it goes to show how far, I mean, people were already thinking about this, 24 years ago, this idea of expanding what a virus is. So, if you think about this, an expanded view of what a virus is, the proteins encoded in the viral genome as well as the sum of the host interactome. And as Michael emphasized, also location, thinking about where these interactions actually occur. So, thinking about viruses, viruses, the small genome size I think is kind of a two-edged sword. Most plant viruses have genomes that have a coding capacity of about 4 to 10 proteins. Some viruses have more, a few have a few less, but that's kind of the ballpark for what a plant virus genome is. And the thing is, the primary functions for plant virus proteins were determined many years ago. If you think about the virus genome sequencing project, it actually began in 1980 with cauliflower mosaic virus, the cauliflower mosaic virus sequence. And if you think about what happened in the 80s through the 90s, basically lots of plant virus genomes were sequenced. And basically what the message was, the common functions. This is what you have. You have a replicase protein, maybe 1 or 2 replicase proteins. A lot of times you have 2. Movement proteins. A lot of viruses have 1 movement protein. Some will have up to 3. Capsid proteins, maybe 1 or 2 capsid proteins. And then you have kind of a smattering of other things, proteases, aphid transmission factors. But the point is that I think we got set on saying, well, okay, this protein is a replicase. But the thing is, as Michael emphasized, plant virus proteins are multifunctional. And we have to start thinking about them a little bit differently. I thought that was interesting. Linda Kinkel was talking about, again, this idea of rethinking how we look at things differently, and I think we need to start looking at viruses differently. I'm going to just talk briefly as an example, the multifunctional nature of the P6 protein of cauliflower mosaic virus. So this is something that we put together. And if you think about this, you have sort of the basic host pathogen things. P6 triggers HR, for example. P6 is also a symptom determinant. We don't really know why P6 causes symptoms yet, so we don't really know the interactors for that. But P6 also does a number of things in the viral infection cycle. It's a virion factory, and I think people are really keying in on this idea that viruses form these virion factories, and they're actually, I would argue that they're kind of like pathogen organelles, and so you think about a chloroplast and what a chloroplast is. Well, we should start to think about, what these pathogen organelles actually are. And P6 is a pathogen organelle. So basically, replication, translation, and virion assembly and virion accumulation occurs within this pathogen organelle, the P6 protein. P6, 20 years ago, was shown to be a translational transactivator. It actually converts a ribosome, a eukaryotic ribosome, into a prokaryotic ribosome. So you can translate polycistronic messages. And there's a series of proteins that have been shown to interact with P6 here. P6 is involved in intracellular movement. Michael showed the interaction of proteins with the cytoskeleton, microtubules. P6 also is involved in microfilament movement. It's a silencing suppressor. Groups have shown that it's a silencing suppressor. And lastly, it's an alteration of host defenses. So you have all of these multiple things that this protein is doing. And in fact, in terms of considering interactomes, we should be thinking about interactomes, again determined by protein-protein interactions, as well as location. Where are these happening? And so you can start to put together interactome maps for, say, intracellular movement. And I'm not going to go into the details, but just to emphasize that we're kind of in the infancy, at least in terms of CAMV, what these interactome maps would actually look like. This would be, basically you have the P6 protein. This is what an inclusion body looks like. P6 carrying all of those virions in that inclusion body, kind of the end result of that virion factory. But then you have these various proteins, host proteins, that interact with P6, shown either genetically or through protein-protein interactions, that may facilitate movement. And what you find is, not too surprisingly, lots of redundancies in terms of function. So the virus has evolved to interact with lots of different proteins, again, to be successful. Now, the thing I would emphasize is that this is just cauliflower mosaic virus. I mean, you could go through, many of you, and we heard talks today that were very similar, Basically, we're looking at this in similar ways. But if you think about this vision of actually kind of expanding what a virus is to include the host proteins, I think it could be very valuable. So, what's the contribution of viruses to the phytobiome? Viruses are ubiquitous in nature, and as Rodrigo showed, some viruses, you have these persistent viruses. They're always found in association with their hosts, pepper and some of the pepper viruses. Other viruses are frequently found in association. I think about barley yellow dwarf in wheat, for example. A large part of the wheat crop is actually infected with barley yellow dwarf. And so when you consider the phytobiome, the contribution of other organisms, should you consider that without barley yellow dwarf or with or both? And then secondly, other viruses may influence the host through infections with fungal endophytes or nematodes. So you have all these different categories as well as bacteriophages. But there's also an opportunity offered through plant virus research. I think if you think about phytobiome research, the goal is to identify microbes that have some beneficial effect to the host. But the other thing about viruses that you have to realize, and I think this is a theme that's coming out over and over again, viruses modulate host defenses. And so, we also have to start thinking about, what's the impact of that? If you have a virus-infected plant, what's it doing in terms of predisposing a plant for a fungal infection or, a bacterial infection or any other pathogen? So, and also, if the goal is to identify microbes that have some beneficial effect to the host, the next question is how? What's the mechanism? And Anna touched on this as well, talking about this. Now, the small genome size of viruses may contribute to mechanistic studies. So you have 4 to 6, or maybe even 10 effector proteins. And Michael, there was one thing he didn't really emphasize in his slides. Viral proteins equal effectors. Every viral protein is essentially an effector protein. So you have maybe 4 to 6, maybe 10 effector proteins for viruses, compared to maybe 30 to 300 effector proteins for other pathogen groups. So we actually may have a manageable system for getting at how viruses, what are the precise interactions that lead to changes in host physiology. And I think that's something that was touched on in a number of talks today. It's a goal that we have. So with that, I'd like to thank the speakers again for, I thought was a really excellent session. And then I also want to thank the APS meeting program who provided funds for the speakers to come here, as well as the Samuel Roberts Noble Foundation and the Widely Prevalent Virus Committee, two groups that have been consistently funding plant virus talks at APS for many years. And with that, I'm going to finish. I'm done. Thank you very much for coming.